Hello friends, welcome to Agrim Medical Concepts. Today we will discuss certain very important MCQs from the topic antimicrobial resistance, and these MCQs are very important for NEET PG entrance exam. So here comes the first question. Just look at this figure. So here our question is, biochemical mechanism of drug resistance are shown in this given below figure. Most important mode of resistance in MRSA is A, B, C or D. So friends, resistance in MRSA, there is methicillin resistant Staphylococcus aureus. So resistance in MRSA occurs mainly because of altered penicillin binding proteins. So here we can see in this figure that option A is talking about the target site modification. There is altered structure of the target. There is altered structure of the penicillin binding proteins. So option A is correct here. And based on the same figure, our next question is, Enterococcus develops resistance to vancomycin by which mechanism? A, B, C or D? So friends, again here vancomycin resistance occurs due to altered binding site whose structure changes. So again our answer is A. And now our next question is, C is the major mechanism of resistance against option A fluoroquinolones, option B aminoglycosides, option C tetracyclines and option D sulfonamides. So here option C is telling us that there is formation of certain enzymes in the bacterial cell which are degrading the antibiotic. So this mechanism is applicable in case of aminoglycosides. Yes, aminoglycosides face resistance because of this enzymatic breakdown. And yes, now the next question based on this figure is, major mechanism of development of resistance against cotrimoxazole is A, B, C or D? So here we should know that cotrimoxazole contains sulfamethoxazole, which is a sulfonamide, and trimethoprim. And resistance to sulfonamides and thus cotrimoxazole occurs due to alternative metabolic pathway. That is, bacteria start utilizing preformed folic acid. So mechanism D is applicable here. There is bypass. It means altered metabolic pathway. And now our next question is, E is the major mode of resistance against tetracyclines, chloramphenicol, ampicillin or ciprofloxacin. So friends, sometimes there is formation of certain efflux pumps in the bacterial cell.
Efflux pumps are transport proteins that expel substances, including antibiotics, from cells, primarily playing a role in drug resistance in bacteria and some eukaryotic organisms. They are found in the cell membrane and help maintain cellular homeostasis by removing unwanted metabolites and toxins.
Hence, the answer in this case will be tetracyclines because of formation of efflux pumps which throw the antibiotic out of the bacterial cell. So friends, here in a nutshell, you must know about certain very important mechanisms which are responsible for the resistance against distinct classes of the antibiotics. So just look at this table. So if we talk about enzymatic breakdown, this is the mechanism of resistance in case of ABC. There is aminoglycosides, beta-lactam antibiotics and chloramphenicol. The next mechanism is decreased permeability.
Reduced permeability, a mechanism of antibiotic resistance, involves a decrease in the ability of antibiotics to enter bacterial cells, limiting their intracellular concentration. This can occur through various means, including changes in the outer membrane, cell wall, or poron channels, all of which affect the entry of antibiotics into the cell. Reduced permeability to antibiotics is a more significant issue in gram-negative bacteria due to their unique outer membrane structure. This membrane acts as a barrier, limiting antibiotic entry, and is particularly evident in gram-negative bacteria like Pseudomonas aeruginosa and Acinetobacter baumannii. Gram-positive versus gram-negative. While gram-positive bacteria, with a thick peptidoglycan layer, can be susceptible to reduced permeability as well, the outer membrane barrier of gram-negative bacteria is a more prominent mechanism of antibiotic resistance.
And this is the mechanism responsible for resistance against aminoglycosides. Next is efflux pumps. Formation of efflux pumps against tetracyclines. Then altered metabolic pathway in case of sulfonamides. And last but not the least, altered target, that is change in the structure of the target. This is seen in case of MRSA, vancomycin and fluoroquinolones. Now the next question is, anaerobic bacteria are intrinsically resistant to beta-lactam antibiotics, aminoglycosides, chloramphenicol or metronidazole. So the answer here is aminoglycosides, because these antibiotics are not active against anaerobic bacteria because their uptake across bacterial cell membranes depends on energy derived from aerobic metabolism. This dependence on aerobic metabolism is the main cause of markedly reduced activity of these agents in areas of low pH and low oxygen tension. For example, abscesses and other infected hypoxic tissues. And now here comes the next question. Mycoplasma is resistant to vancomycin, doxycycline, moxifloxacin or azithromycin. So basically here vancomycin is the answer. So well, actually this mycoplasma lacks cell wall. Thus any drug acting by inhibition of cell wall synthesis, for example vancomycin and other beta lactam antibiotics, is likely to be ineffective. So the answer here will be vancomycin. And now the next question is, multiple drug resistance is transferred through transduction, transformation, conjugation, mutation. So basically multiple drug resistance is transferred through plasmids, mostly by conjugation. The well-known examples are MRSA and VRE, that is vancomycin resistant enterococcus, and also Mycobacterium tuberculosis. And here is the next question. Most common mechanism for transfer of resistance in Staphylococcus aureus is conjugation, transduction, transformation and mutation. So basically you have to heed that transduction is particularly important in transfer of resistance among staphylococci. So the correct answer is B, transduction. However, already we have discussed that multi-drug resistance is transferred by conjugation, for example MRSA. And here comes the next question. Elaboration of inactivating enzymes are the important mechanism of drug resistance among all of the following antibiotics except options are quinolones, penicillins, chloramphenicol and aminoglycosides. Basically, here the resistance against quinolones or fluoroquinolones is mediated by mutation in the DNA gyrase enzyme. So the answer here will be A, quinolones. So friends, please pay special attention to all these questions because these are very important for NEET PG examination. And if you have any queries, you can write it down in the comment section below. So soon I will be back with more questions based on this antimicrobial resistance and antimicrobial therapy. Thanks.